The ability of a microbe to cause disease is termed pathogenicity. The degree of pathogenicity, basically the ability to infect and cause disease, is called virulence. You can see on this image right here that we can classify or scale organisms based on their virulence, so how easily they can actually infect and cause disease. You can see that plague is near the top of this, so it's very virulent or more virulent than something like thrush or lactobacilli. The pathogen traits that interact with the host to enable entry, attachment, and or escape host defenses, these are all going to be things that affect the virulence, and they're going to be what we call virulence factors. We've talked about some of those already. Virulence factors, they can include things like adhesion factors, affecting the ability of that microbe to actually attach and stick to the host. There is some specificity there. There's also some proteins that are involved in biofilm formation. So those can affect virulence if it's very easy for the biofilm to become established. There are a number of extracellular enzymes that can affect the ability of a microorganism to infect or to enter the body. Here we have a few examples on this slide. One example is that bacteria might come into contact with a human surface, like epithelial cells, and then those enzymes might help to dissolve the materials that are holding the cells together. And in this way, the bacteria are creating a little entry site or a little pore for them to enter the body. Another example is that bacteria might have special enzymes that help them to evade our host defenses. And here we can see that we might have bacteria that produce coagulase, which is going to help a clot to form. And then later, they actually dissolve that. And again, they can enter the bloodstream in this way, like you see in this image here. There are a number of toxins that can be produced by microorganisms. Toxins can be considered exotoxins. These would be if they are ones that the microbe secretes. They can be considered endotoxins. Both of these can actually cause quite a bit of problem, and they can help with virulence. And then another one is that we can have what's called antiphagocytic factors. These affect the ability of the host to actually engulf and destroy that bacteria after it has entered the body. There's a couple different ways that this can happen, and they're illustrated in the images right here. It might be that the bacteria has a particular capsule around them that prevents the host cell from actually being able to really break it down. We could also have an example where the capsule protects the bacteria, and then the bacteria, even if they're engulfed, they can continue to reproduce inside of the cell that did engulf them. So both of those, again, are a way to kind of get around the defenses of the host cell. The stages of infectious diseases fall into 5 different categories, and those 5 categories are what we're seeing in this graph representation. If you look at the y-axis, this is measuring the number of organisms that are present, or the number of microbes that are present, and then also the intensity of the signs or intensity of the symptoms. So how bad it actually is. After the initial infection, there is an incubation period. During this incubation period, we don't really see any signs or symptoms. We might just have a little bit of replication or multiplication of the pathogen. During the next stage, the prodromal period, this is going to be where we start to feel some of the effects. The microbe becomes more abundant. And then when we get to the 3rd stage, we have the full-blown illness, and we start to feel the full force of all of those different signs and symptoms that that particular infectious disease is able to cause. We also have the highest numbers of the microbe when we're looking at the illness stage. The next stage is going to be the decline. We're going to start to see fewer microbes. This is because the body's ways of actually fighting that particular microorganism are really kicking into gear, and they're starting to cause it to decline or become less. And we're also going to see some recovery in the symptoms where we're not feeling as many of them. And then the last stage is called convalescence, and at this point there are no signs and symptoms remaining in the individual. So that would really be kind of the recovery, fully recovered point. Here is a look at some of the incubation periods for selected infectious diseases. Now, the incubation period is the period where we weren't really feeling the effects of the disease, which means that we're probably still going about our everyday lives, we're still going to work, we're still going to school, and we're still coming in contact with a lot of people. And so you can see that there is quite a range here. In some cases, it's years before we actually start to move into the second stage where we start to feel some of the symptoms. And the longer the incubation period, sometimes the easier for it to spread, because again, we are coming into contact with a lot of other individuals, which is meaning that we are serving as essentially a human carrier and exposing other individuals to ourselves, which are now the reservoir. Now, after the infectious process, the pathogen has to move out of the host if it's going to infect another individual. Pathogens leave infected patients through what we call portals of exit. This slide represents the portals of exit. A lot of them are going to be the exact same as the portals of entry that we looked at earlier. So we do have natural body openings that can serve as portals of exit. There are different body fluids that can come out of those. And then we also have injuries that can also cause a release of the pathogens from our body and into the environment where other individuals might become infected when they come into contact with them. If we talk about how infectious diseases are transmitted, transmission from a reservoir or a portal of exit to another host portal of entry. That's what transmission means. There's a number of ways that this can happen. One of them is just contact transmission. You can see several examples of contact transmission on this slide here. We can have contact where it's our skin and another skin, so that could be something like shaking of hands. We could have sweaty towels and things like that. So if we come into contact with that, or if we are drinking from the same bottle as someone else or sharing cups. You've got the sneezing. So fluids coming out through sneezing or coughing is also an example of contact transmission. If we come into contact with that, we can become infected potentially. There's also what we call vehicle transmission. And so this is going to be spread of pathogens via air, drinking water, and food. And you can see that in a market such as right here, you can have the spread of pathogens in those food sources. There's many foodborne illnesses or drinking water-borne illnesses. We can get the spread of pathogens in that way. And then another one is going to be vector transmission. When we're talking about vector transmission, we're talking about some type of live organism that is transmitting that pathogen from the reservoir to the host. So this might be through an arthropod, an insect. It could be a tick feeding on one organism and then another. It could be a mosquito feeding on one organism and then another. So that's vector transmission. The classification of infectious diseases can be based on a number of different characteristics. Some of those characteristics could be the body system that's affected, how long the disease actually persists or lasts. It could be classified according to its severity. In some cases, the taxonomic group, so what organisms are responsible for the diseases and grouping them in that fashion. We can also group them according to whether they are communicable disease or not. All of these characteristics of diseases are used pretty regularly to classify different diseases. In this table, it's just showing you a representative list of some types of diseases, or really terms that are used to describe diseases. For example, we can have contagious diseases, we can have local infections and systemic infections. Local infections would affect just a small area. Systemic is going to be a widespread infection. Another primary infection would be the initial infection. Secondary infections are going to be other infections that happen because of the first infection. So that could be something that is due to opportunistic pathogens becoming more abundant. Epidemiology is the study of where and why diseases occur, and then also how they're transmitted within populations. When we're talking about human diseases, this is certainly something that is studied a lot. Epidemiology usually results in the construction of a lot of graphs and pictures to represent both incidence of a disease and then also prevalence of a disease. The incidence is going to be the number of new cases, and then the prevalence is going to be the total number of cases that we have. Here's a representation of cases that we might have per 100,000 individuals in a population. Look at the United States. You can see that in this particular image, the prevalence, how common this disease is quite great in the Northwest. So in the North Midwest, really, in this diagram. And then we see a lot less of it when we're looking at the eastern side of the United States. We can look at this in regard to times of month, so whether it is seasonal or not. In the first graph that we see here, we can see that there are certain seasons where it's quite abundant. We can also look at this in regard to how prevalent it is in different age groups. In this case, we can see that we have a lot greater number of deaths when we're talking about older individuals. In this picture right here, we can classify infectious diseases as endemic. Those would be ones that just pretty much always occur in that particular region, and we have a pretty standard number of new infections all the time. Sporadic means they just pop out kind of randomly. Epidemic means there's a sudden outburst of that particular disease in a particular location, so an increase in the incidence. And then pandemic means that we've got lots of increases in the infection in many different areas. Here's another way to look at it. You can see that when we're talking about an epidemic is an unusual number of cases. Just because you have a high number of cases does not necessarily mean that you're going to have an epidemic. You can see that in the disease being shown up here at the top, this is going to be the expected number of cases being shown by the blue line. And that one, we always have a high number of cases. And so even though there's a lot of cases, it's not considered an epidemic because it's a pretty even number throughout many years. In the one at the bottom, we tend to have fewer cases of it. And you can see that there are certain times when we tend to have a bunch of cases reported. So you can see really two different epidemics being shown for that particular disease. There are a number of ways that epidemiology is studied. Epidemiology can be studied descriptively. Descriptive epidemiology is going to be a careful collection and tabulation of data concerning the disease. In this case, there might be a map. We might be trying to pinpoint exactly where the new cases are developing. We might be able to identify an index case. The index case is the first case that occurred, and then we might be able to see how it has spread from that particular location. In this picture right here, we see some clumping of it around a particular well or water source. And so in this case, that might be identified as the initial location where this developed. And then another way to study epidemiology is analytically. Analytical epidemiology is going to be an in-depth investigation of the disease. And in this case, it's an analysis of data from descriptive epidemiological studies. This is going to include really looking at everything. This is looking at the probable cause, this is looking at the mode of transmission, and then also hopefully looking at the means of prevention of that particular disease. Experimental epidemiology involves testing of a hypothesis, trying to really prove the cause of the disease, and eventually getting into Koch's postulates to really conclusively determine that this is the pathogen that causes that specific disease. The last thing to talk about here is what we call hospital epidemiology. There are a number of infections or diseases that are acquired by patients and healthcare workers when they're in some type of healthcare setting. This could be actually being admitted to the hospital. It could be working at the hospital. This could be going to a doctor's appointment, waiting in a waiting room. All of that is related to a healthcare facility of some type. So that's what we're talking about here. When you go to those places, you can be exposed to pathogens that are there, and you can potentially become infected with them. As you can see from this infographic right here, that 1.8 million people per year acquire an infection during a hospital stay. So that's one of the examples that we're talking about here. You're admitted to the hospital and you obtain an infection that you didn't have when you went into the hospital. So there are ways to help lower these numbers. Is certainly something that has to be paid attention to a lot by healthcare workers. Ways to help prevent these types of infections would be hand washing, proper cleaning of instruments, cleaning of floors and restroom facilities, bed sheets. All of those things are going to be important for helping to prevent hospital-acquired infections.